And thank you very much for having me here. MiFoBio is really a unique place. You know, I think I was already in the second iteration of MiFoBio, and it's always an amazing place of inspiration and so much creativity talking about imaging. So thank you very much for having me here today. So I need to go through a few disclosures. Apparently, my opinions are not completely free of conflicts, but I'll do my best. Anyway, what I care about is this giant machine, which is really the different proteins that are going to be needed to direct a polymerase to transcribe a gene. About 10% of our genome, of our proteins, are dedicated to this process. And for me, it's really a giant combinatorial machine that is devoted to dynamic adaptation, right? You need to do the right thing at the right time. We know biochemically most of the players, but we have a fairly static view of it. And what is missing is really an understanding of how dynamically this thing is regulated, right? But for me, it's a playground to study regulation and to study a very complex machine using microscopes, which is kind of fun. So the technique I'm going to use is single particle tracking, and this is a technique that was developed when my lab was in Paris by Ignacio Izedin. And I must disclose that everything I'm going to tell you today was proven to become obsolete by what Ignacio and his group showed 2 hours ago. And so I think tomorrow they will show it again. They are now using these events cameras that that are going to completely add a new page to this technology. But I'll let you go tomorrow to their show at 4 p.m. So basically, we see a single protein, and we track it either at 100 images per second or 2 images per second, depending on what we want to see. And out of this, we are going to see how this molecule is moving within the nucleoplasm of a cell, whether it's going to bind to something. Here, I care about binding to DNA, but it could be a membrane, it could be the cytoskeleton. And so what we are really extracting from these movies is a concentration. I mean, we can see if there is more or less, molecules. It's relative because we don't see all of them. It's sparse labeling. A diffusion coefficient, diffusion mode, which is something Ignacio really worked a lot, how these molecules are moving in space. How many of them are bound to something? When they bind, for how long do they bind? And all these softwares we have been developing for the last 15 years are going to be completely obsolete because, again, Ignacio and his group are bringing a software, a hardware technique, that is going to completely solve the problem of tracking. But I let you tell about this. So today I will broadly touch on a few overarching concepts. And it's really the idea that the dynamics of the molecular complexes that we believe to be stables, right? When I started working on this field, we really believed that to trigger the transcription on a particular gene, which is a program that is going sometime to last for the life of an organism, we needed to have stable complexes that are going to stay in place for hours. And so we are going to see how dynamic they are and how it changes our view of regulation. And so we are going to talk about transient interaction and how they are dominating things. We are going to, a general theme of what of the different examples I'm going to illustrate today is that we are going to care more about the binding frequency, how often molecules are binding, so basically the K on, than the binding time, which is a K off. And we are going to see that in transcription regulation, at least, there is very little, very stable complexes, with very strong interactions. And it's very often systems where molecules are constantly interacting and breaking apart. And it's really a K-on regulation. And so one of the first illustrations of this really comes from work that was done in my lab a few years ago now, working on a specific transcription factor. This is a protein that binds DNA, bicoid. Bicoid is an important transcription factor in Drosophila development. It's going to be present in the developing embryo at very high concentrations in the anterior and at very low concentrations in the posterior. And really, it's going to dictate where you put the head of an embryo and where you put the tail of an embryo, of a fly. And so the model that was accepted when we started this work, when Mustafa started this work, is really to acknowledge that at high concentrations of bicoid, different sites on the DNA are going to be occupied. And so because there is a protein-protein interaction, two molecules that are binding to adjacent sites on the DNA are going to interact with this DNA, but are also going to interact through a protein-protein interaction. And this co-occupation of these two sites is going to create a stable interaction that is going to bind for a very long time. And so at low concentrations, you don't have enough molecules to bind the two sites at the same time. You either bind one or the other. And therefore, you don't have this protein-protein interaction that will stabilize the interaction, and you bind for less time. And so we said, great, you know, we have now this technique. We are going to label single molecules of this transcription factor in a developing fly embryo. At the time, we built one of these lattice light sheets that was developed by Eric, and we started measuring bicoid in the developing embryo. And so here you see these blinking molecules, you know, that are not really blinking. They are moving around and they are focalizing and defocalizing. But what you can recognize here is none of them is really immobile. Right? We don't see very long bindings on chromatin. We don't see mobilizations. This is because the longest events we could record were less than a second in time. And so this is a transcription factor that is binding in the second range on DNA, and yet it is triggering a whole program of transcription, a whole program of development. And so we had to now understand how is this going to regulate transcription if it's binding for such short amounts of time. And so from this, we evolved, you know, from the model I showed you where we believed these molecules would stabilize to a model where at high concentrations of bicoid, you have a sufficient amount of molecules that the DNA element is going to be constantly occupied by new molecules of bicoid. And yes, of course, each of them is binding for a very short amount of time, but they are constantly replacing each other, and this is where you get the activity from. At low concentrations, you have a few sporadic binding events that are going to be inconsequential, right? And this is really one of the examples, you know, of us going to, instead of having a regulation at the K-off, you know, we don't care anymore how long you are going to stay bound to a particular site, but we really care how often you are going to visit these sites. So another important example here that is illustrating this, this point is Anders Hansen, when he was in the lab, is now, so Mustafa is now at UPenn, Anders is now at MIT. And so when he was a postdoc in the lab, he started caring about this protein called CTCF. CTCF is another DNA binding protein. And so again, he started labeling CTCF and measuring how CTCF was moving around. And when you see this trace, you know, for someone who is really used to see single particle tracking, there is something weird is that it looks like it's moving. It's clearly not binding on DNA because it's moving more than that. But it looks like it's trapped in a small domain. And so Anders was a little bit puzzled by this. A long story, very, very, very short. He ended up zeroing down on this domain of CTCF, this RNA binding region of CTCF that he mutated and by genome editing replaced by 3HA. So basically he removed this RNA binding region. And by removing this RNA binding region, he could transform CTCF doing this weird movement. And this weird movement, this is something Ignacio developed in the lab, is something we can quantify as this tendency of anisotropy movement. You know, when you see the choreography of a particular protein, you see, is it moving randomly in all directions, or if it's trapped in a domain, or if it's doing, or if it's constrained in the space it's exploring, we see this very strong anisotropy in exploration. And this was due to this RNA binding region. I told you before that we can measure using our single tracking assay all the parameters of mass action, K-on and K-off. And so what Anders was able to measure here is that this RNA binding region, you know, and this trapping this molecule in these small regions is really not affecting the K-off. We don't affect how long CTCF is going to bind when it interacts with DNA. But what it is changing, and by a factor of 2.5-fold, is how often CTCF is going to bind to this target. And this is just by trapping the molecule in small regions where you are de facto going to increase the local concentration, the apparent concentration, and change the frequency of binding events. So again, a regulation of K-on and not a regulation of K-off, of the on rate. So, what are the concepts here? The concept here is, this kind of K-on and how we are going to regulate it. We are going to see that it's really the result of weak interactions that are essential for this rapid dynamic, right? If we have something that is binding for a very, very long amount of time, it's going to be extremely hard to regulate. Transcription is something that we want to be able to regulate on and off. And so if you want to be able to turn it off, you cannot use very strong interactions. So that's very useful for the way transcription is regulated. And so then we can see a trade-off in between strengths and numbers. Of course, with a single molecule and a very strong interaction, you can achieve full occupancy of a site. But we can do the same with multitude of weak interactions, that are by numbers going to give to a certain strength and to a certain occupation of sites. At the end, mass action can be harnessed by local enrichments. And this is really what I'm illustrating, with this graph, saying that if you don't bind for very long, you can compensate by binding often. And how do you bind often? There is different strategies that we have seen over the years. This is an image made by Ignacio and Ibrahim when they were in the lab, where they measure the concentration of RNA-pol2, and you see these accumulations. And so, of course, if you bring more molecules locally, you are going to bind more often, and this is a way to regulate K-on. And what Anders saw, and Ignacio saw too, is you can regulate, you can increase the K-on by diminishing the space. You still have a single molecule. If this molecule is navigating in a space that is confined, you are de facto increasing the concentration, and you are increasing the K-on. So, so far what I really want to stress is to pay a little bit of attention, and this is really something that has been triggered by this imaging of single molecules. Pay a lot more attention, to the on rate, how often you bind compared to how strong you bind, which is really our textbook vision of regulation of macromolecular complexes in cells. So now I'm going to pivot a little bit and tell you about other experiments, and we are going to go to different pieces of data illustrating how these interactions are distributed in the different factors that we are studying. And for this, I'm moving to a cofactor, this giant protein P300 that Jonathan Carr and Jack Ferry decided to tackle when they were in the lab. And this is a monster protein that is going to, that was believed to interact with chromatin with its core here, and had this C-terminal region and this N-terminal region that contain all these domains, and these domains are transcription factor interaction domains. So we had a core that was interacting with chromatin. We had two domains on the sides of the protein that are supposed to interact with transcription factor domains. And what these two really decided to do is start understanding how this protein is negotiating chromatin, how this protein is binding to chromatin. And here again, I'm drawing a long story short. But if we look at the fraction bound here of wild type P300, or if we delete, for example, all these transcription factor binding domains, we see that this is really responsible for it. What I'm not showing here is that the core was not important. So this domain that was annotated to bind chromatin was really not the domain important to immobilize this protein on chromatin. So we see that we need, if we remove all these transcription factor binding domains, the protein doesn't bind to chromatin anymore. If we remove them one by one, we have a minor effect. And if we express them one by one, and label them with a fluorophore and track them, they have also a minor effect interacting with chromatin. And so this is a point that is very important to illustrate here, is this is a communal effect in between all these domains, and one of them is not sufficient to bind efficiently. Altogether, they bind very well, but you need this multitude of domains to act together, and so to weakly bind, but weakly bind several times to different factors. And so now, it's a very, very good analogy to this Velcro effect I gave you before. It is a protein with long IDRs, that is binding to different transcription factors. These transcription factors are going to bind DNA. And then you have these multiple weak interactions with DNA, with chromatin, that are going to be essential for this protein to bind DNA, right? So it's a binding that is protein-protein mediated here, because you have an intermediate binding to the DNA, but is distributed over different domains, over different proteins. And each of them is weak. And you only come to the strength of interactions through a multitude of binding domains. So another piece of data I wanted to show you with another family of proteins. Now I'm coming to another family of transcription factors, and these are factors that are essential for hypoxia in cells. When you are lacking oxygen, these transcription factors are going to be activated. And so we have two factors. We have if1 alpha and if2 alpha. And so they have nearly identical DNA binding domains. And so we had envisioned that these two factors would bind DNA in very similar ways, yet functionally they have many different functions. Actually, one of them is an oncogene, one of them is an anti-oncogene. And so they bind to the same, they have the same extremely similar DNA binding domains. So we believe they bind to, they bind to the same sequence. But they have these different functions. And so we decided to try to understand how they are navigating chromatin, how they bind DNA, using our techniques of single particle tracking. And so Yu Chen in the lab, who published this work now a few years ago, realized that these factors are typical factors. They have a DNA binding domain and they have an activation domain, this low complexity domain, this IDR. And so she labels them with halo tags to be able to track them. And so she realized that if 2 alpha has a bound fraction of about 35 to 40%, so it's really a lot of it is bound to chromatin. But if 1 alpha only has about 15 to 20% bound to DNA. So the binding to DNA is completely different. And so we were really expecting this to be mediated by the DNA binding domain. I already primed you saying that they're extremely similar. But what she realized is that this binding property, how much of the protein is bound to chromatin, is not mediated by the DNA binding domain, but is really a feature of the IDR of the activation domain, which is a protein-protein interaction domain. And so here what we are realizing is that transcription factors, they don't negotiate chromatin and DNA just interacting with the DNA sequence, but there is also a myriad of protein-protein interactions that are probably coded by the IDR, by other protein-protein interaction domains, and this is really what we care when we think about transcription regulation. And this was really the take-home message of this paper. And so when we think about the transcription factor, we very often think it's of binding to DNA, to binding to a specific DNA sequence. But this is really not the right vision. The right vision is to think about chromatin binding in general. And yes, you are going to end up binding in a DNA, at a particular cis-acting element, a DNA sequence, but this DNA sequence is decorated by a tremendous amount of proteins in chromatin, and there is a tremendous amount of protein-protein interactions that are going to be important for these binding events. And so this is what really I'm illustrating here. It takes a village to load a protein on DNA, navigating the nucleoplasm and chromatin. And so from a diffusing transacting factor, you know, our transcription factor navigating, we very often call them transacting factors, we are going to have a myriad of interactions that are going to end up lodging the DNA at its right binding sequence. And so IDR, weak interactions, you know, in between low complexity domains, more stochastic protein-protein interactions through the folded domains of these proteins, non-specific interaction with DNA, where we know that these transcription factors are going to bind DNA in a non-specific manner and slide on it to end up, you know, binding to the DNA. The beauty of this kind of models is that all these interactions are weak. All these interactions are very transient, and so it gives rise to a system that is, you need a multitude of interactions, they are all weak, they all can be displaced, and so you have a system that you can regulate. So, when you realize that you have all these protein-protein interactions that are important, one thing you realize when you look at this is that we badly need tools to measure protein-protein interactions, right? When I show you a transcription factor immobilized on chromatin, immobilized on DNA, I am not telling you what are the partners. I am not telling you which kind of protein it's interacting with. And so Thomas Graham, a postdoc in the lab, decided to tackle this problem. And to tackle this problem, he came back to this monster system that is really a guinea pig for us and an amazing tool. Laurent Elliot is working on this too. Ignacio is working on this too. This is a positive transcription elongation factor B. This is a kinase that is important for transcription regulation. The only thing we care about here is this is a maze. This is a maze because this factor, 2 proteins, is present in 1, 2, 3, 4, 5 different complexes with 5 different functions, with 5 different regulations. And so if I label one of the components of PTFB, I have no idea what these proteins are doing in a cell, because when I label them and I look at them at the single molecule level, there is nothing that looks more than a single protein than another single protein. And I have no idea what kind of complex they are engaged in. And so Thomas, when he was in the lab, one day was doing an experiment where he had 2 proteins. One was labeled with GFX 650, and the other one was labeled with the Genilia Farm 549 dye. And so Ignacio was looking in the green channel to image one of the proteins, and then he was bleaching the other. He decided to bleach the other protein and to image this one better, right? He had a little bit of leak, or he didn't. I mean, I don't know what he was thinking. I'm not sure he knows himself. But what he realized is that after doing this, when he images green molecules, some of the bleached ones, some of the GF650 reappeared. And instead of saying, okay, that's weird, what Thomas did is he realized that when he sends a GFX 650 dye to the dark state, when he bleaches these molecules and they disappear, they can come back, and they can come back as a function of the presence of a GF549. And when we excite the GF549 with a 561 laser, some of these molecules are going to become fluorescent again. Right? This is something we knew we could do with a UV laser at 4 or 5 nanometers, but we didn't know we could do it with a 561 laser, and especially not as a function of the presence of another dye. So all of a sudden, he had a tool to interrogate whether his molecules were alone or whether these molecules were in the presence of a partner, and so he could interrogate the proximity of these 2 dyes in cells and reactivate fluorophores as a function of the proximity of another molecule. So this is really what we're doing in these experiments. Of course, the movie I've been troubleshooting the whole afternoon is not going to play. Oh, it does play. So basically what we do, we bleach, and then we reactivate in green, then we bleach again, then we reactivate with a UV, and again and again and again. And we constantly compare how many molecules we are going to reactivate from the dark state with the UV laser, and how many molecules we are going to reactivate with a green laser. Right? And this is going to give us an idea of the proximity of another dye, because we know the green reactivation is going to be function of the proximity of the other dye. And so he tested this with this molecule cyclin T1, where he labeled part of PTFB, and then he measured the interaction with another complex, which is protein exim-1. And so these are the two partners that he decided to label. And we know that it's a system that is controlled. We have drugs that are going to dissociate the complex. We have cellular conditions where this complex is going to be dissociated. And this is how we read the results. We always compare the direct reactivation using the UV laser. And so you see that this molecule has, in its native way, if we look at all cyclin T1, we see a small population that is diffusing relatively slow, and another population that is diffusing a little bit faster, right? So we have two diffusion speeds, and so you can imagine that this is the case. We have this protein is engaged in many different complexes. So now when we do the PAP as a proximity-assisted photoactivation and reactivate with a green laser, we get a completely different distribution of these molecules. Most of them are diffusing slow, and a few of them are diffusing fast. And this is really telling us that the hexim cyclin T1 complex, this large complex that contains an RNA also, is this small population, this population that is diffusing slower. More interestingly, doing these experiments, we can also time things, and basically we can use a drug at time 0, and we can measure this kind of peak, how it's evolving over time, and we can measure how these two proteins are dissociating after treatment here with a drug that is going to dissociate this complex. So this is really a game changer for us, because now we can look at the dynamic of this complex, and I can identify which peak in this diffusion coefficient is what kind of complex engaged with what kind of proteins. And as I'm going to show you in the next slides, this is really a game changer for the kind of research we are doing. So at the same time the lab was doing this, I also started a company, and in this company we decided to use these single particle tracking techniques to follow single proteins, and to follow single proteins to see if we can modulate the dynamics of these single molecules, these single proteins, with drugs, and whether we can discover drugs using our microscopes, right? So basically drug screening, screening libraries of small molecules. So in order to do this, Ignacio, in the technique he had developed in the lab, we were either using high-low illumination, which gave us a decent field of view, but with a oil immersion objective and a relatively small field of view. And so at Acon, the team decided to adopt a new technology that was an idea of Xavier DARZACQ at the time, that we call oblique line scan. And so we didn't invent anything. We used a trick that was known by many. We create a light sheet, and this light sheet, we are going to scan it back and forth in the sample. So it's coming from a single objective. And then we are going to read a single line of pixels out of the camera synchronized with the movement of this light sheet. And this is great because now we can work with a water immersion objective, 40x, and we can obtain the same amount of data we were obtaining, on a single cell, but we can now obtaining on about 40 cells. So we have massively enlarged our field of view. We get a very flat illumination and a very good signal to noise, so we can go to very high densities of single molecules. And so this was a game changer for us, and it's really the technology that enabled Acon Therapeutics to go after drug screening. And so what you are seeing here is an experiment that was not done in a day in this slide, but we cannot now do in a day, screening about 300,000 wells a day. And so the result you get out of this is a few days of imaging, is you are going to test 300,000 small molecules, from a library of small molecules from a chemical library. And so repetitively, we are imaging a negative control, something that is not going to change the dynamic of our protein. We are going to image a positive control that we know is changing the dynamic of our molecules. And we are looking for hits that are new molecules that weren't known before from our libraries that are going to modify the dynamics of this complex. And so here I'm giving an example of a typical screen where we see these outliers. Of course, these molecules need to be optimized, to create a drug, but this is basically our drug discovery pipeline today at Acon. 300,000 wells a day, a petabyte of data generated every day. You can imagine the problems to analyze, and of course, we gave up on storage. So interestingly, you can see how these molecules, are going to change the dynamics of our protein of interest as a function of the concentration, which is going to be really important. And then we can compare it to some biochemical activity of cellular target engagement with these factors. Something I would like to point here is that being able to do this in live cells and using single molecule tracking is giving us, of course, the information of what kind of population of the protein of interest we are modulating. Here we can see that we are with the drug, with this Werner inhibitor, which is a helicase inhibitor that we discovered, is going to modulate, the diffusion coefficient, creating this low population of Werner. But you can see how fast in time this is working, and how this peak is going to grow as a function of time. You can see that it's a very, very rapid effect. And then if we compare to other techniques, characterizing these molecules, we can see that over time we are going to degrade the molecule, but not in the same dynamics as we are changing the diffusion of these particles. And it's really an amazing tool to discover new biology, to characterize small molecules, and hopefully to discover new drugs. Only the future will tell us, if we get these drugs approved. These have extremely high potential to treat different cancers. So we got very excited by this result, and so we brought these microscopes back to the lab, and we started building our own OLSs in the lab to gain the same amount, of larger fields of view and measuring more data. And so these are now experiments that are done again in the lab. We are out of the company, and Abrar decided, can we measure this IDR interaction? So I told you before, I gave you a hint that these low complexity domains that are present in these transcription factors that I care about are important for protein-protein interactions. We know that these protein-protein interactions are extremely weak. Can we use this new technique, PAPA, to measure these weak and transient protein-protein interactions in cells? And so Abrar decided to do this using a model transcription factor that we really care about, because my colleague Bob Tijan has been working on it for now 50 years. He's the one who discovered it. It's the first transcription factor that Tish discovered, the first transcription factor that anyone discovered, the first protein binding DNA, in mammalian cells. And so Abrar decided to study this. So SP1 has a DNA binding domain, 4 zinc fingers. It has also a long, low complexity domain that contain aromatic charges, aromatic amino acids and charged amino acids. And so he decided to study this. We know that SP1 interacts with itself, and so he started by asking the question, labeling one with halo tag and another one with a snap tag, and measuring protein-protein interactions with SP1 with itself, and using this technique, PAPA. So first of all, yes, he's able to measure interactions with PAPA. We can see here the violet reactivation compared to the green reactivations, and you can see that compared to a protein that is expressed at a high concentration in cells, we get far more. And this is really a measure of this interaction of these molecules together. So SP1 can interact with itself, and we can measure it with this technique. And when these molecules interact, we can see that there is an enrichment in the population that is bound to chromatin, and a decrease on the populations that are diffusing. And so it's really the population of SP1 that is binding to chromatin. So this is something that we are now able to measure. And so what Abrar decided to do is to, again, ask this very question, you know, what is the involvement of the DNA binding domain? And what is the involvement on this IDR? And so he decided to remove the IDR, so just express the DNA binding domain, or just express the DNA binding domain with a random sequence, you know, low complexity sequence, but is completely unrelated to SP1, and try to understand what happens. So 50 years ago, in the TIJ lab, you know, my collaborator Bob Tijan, they had proven that the DNA binding domain alone can interact with DNA in vitro and really bind to a very specific region on a promoter of a gene. You have a really precise band, and they even did electron microscopy showing dimers of SP1, now the full-length SP1 with IDR, interacting with each other and interacting with DNA. So we were really sure that the DNA binding domain alone had binding activity to DNA. The problem is in cells, we don't see that. In cells, the DNA binding domain alone of SP1 by FRAP, by single particle tracking, or by SP1 interactions really doesn't interact with itself, doesn't interact with chromatin anymore. And so what is really happening here? So at the same time, Abrar decided to do the reverse experiment where he now expresses low complexity domain alone, since the DNA binding domain is not sufficient in cells to bind to chromatin. What about the activation domain, the low complexity domain alone? And he did two constructs here. He did a construct where he removes the DNA binding domain, and he did a construct where he left a small piece of the DNA binding domain, and this small piece has only one zinc finger. And this is sufficient to confer non-specific interaction with DNA, but not sufficient in vitro to bind to your sequence of interest. And so here, the low complexity domain alone is not sufficient for binding. You might see a little bit of residual binding. Interestingly, this very, very weak mutant, you know, that is nearly not binding to DNA anymore, just a little bit of non-specific binding is sufficient for a little bit of interaction with chromatin and a little bit of SP1 interactions. So that was starting to be interesting. You know, what we're starting to see here is we need both. They both have very weak activities, and they're not sufficient to bring your protein to the right place. Then Abrar studied, you know, the interaction of SP1 with other transcription factors. And I'm adding this slide to remind you that the system is far more complex than SP1 interactions. But you have a myriad of different transcription factors. OC1, for example, and SP1 have a tendency to interact together, and they are going to favor an interaction with chromatin when they interact together. And some of the factors have either weak or transient interactions with SP1. And so we have a different zoology of interactions. So what is happening on DNA? What is happening at the promoter? The way we are seeing it right now is this myriad of interactions, you know, with different transcription factors that are going to weakly interact with DNA, but that are not able to interact with DNA with their DNA binding domain alone. And this interaction is going to be assisted by a myriad of protein-protein interactions mediated by the IDRs. And this is really what's going to help, you know, having this Velcro effect, these multiple grafting points at low affinities that are going to allow these proteins to bind. So I'm reminding you here that the DNA binding domains alone are not sufficient to bind, and that this is really not working. The low complexity domains alone are not sufficient to bind. They interact a little bit with each other, maybe a little bit with chromatin, but not very efficiently. And it's really the mix of everything that is going to give you the sufficient energy, you know, to bring these molecules efficiently to chromatin. So I'm going back, you know, to this model. You know, it takes a village to bring a protein to DNA, and I gave you some illustration of these protein-protein interactions that are important, these protein-DNA interactions that are important too. And so this is important for us as biologists, studying transcription regulation, because now we have a multitude of interactions that are acting together. This is interesting in terms of regulation. This is interesting if you are building a model of how transcription is regulated. It is not very amenable to our classical way of studying biology with a reductionistic approach, where we want to have a domain that we can remove, and the domain is causative for everything, or we want a single protein that is going to be causative for a lot of the things we are studying. Here we need to recognize that we have, it's a group effect, and these molecules are working together with others, and this is not going to work in isolation, and so it's very difficult to study a single one of them in isolation. So with this, I will stop my talk. Of course, I need to acknowledge another village, which are all the people working in the lab. I presented work from all these guys, that are now in different places. Thomas, now at John Hopkins, is starting his lab and hiring people. If some of you are interested in following on these PAPA techniques, different papers I cited today, and I will take some questions. Thank you.
Thank you, Xavier, for this wonderful talk.
Hi. Sorry, is it? Yeah, a beautiful talk, Xavier. I'm very interested in the interaction between the two fluorophores that you saw for the PAPA method. So now it's almost 20 years ago. We saw a very similar interaction between Psi3 and Psi5 in the laboratory of Zhao Zhuang. And I think that what you're seeing here is really exactly the same interaction. I looked into the paper that. So just to reiterate, if you have a red fluorophore, a red cyanine that's going into a dark state, you have a nearby green fluorophore, if you excite the green, you can cause the red to go back into a fluorescent state. And what we additionally saw is if you swap the green fluorophore for a blue or a violet, you can also recapitulate that interaction. So I think that you could actually apply that for, you could have multiplexed detection in this way. But I know that you're doing it in live cells, but we put Psi5 into live cells without the addition of any additional thiol, and the intracellular cysteine is sufficient to recapitulate that dark state of Psi5. So maybe that's what you're seeing.
I totally agree. There is a lot of similarities in between these two systems. We still don't understand how it works. We have no idea what is the mechanism. At some point, it is clearly not a direct interaction between the two fluorophores.
Yeah.
There is no direct transfer of energy. And so we thought maybe there is an oxygen reactive or something like this that is going to diffuse. It works at 20 nanometers, right? 20 nanometers away, we still have this system working.
But
so we tested oxygen reactives, and it's clearly not the case. It works with some dyes, with some others it doesn't. So I agree with you. There is a lot of different options, to make it work with different fluorophores. I would love for photophysicists to go after that, because it would be much, much easier to use a principle that is understood to measure protein-protein interactions than to say, hey, we characterize this, we don't know how it works, but it works.
I think it's super nice, and I don't know if you've done it yet, but exchanging that green fluorophore for a blue or a violet really could let you do multiplexing in this experiment.
So we saw this, yes, it's work with violet dyes. The thing is, so far, we are using the violet direct reactivation for a control. We like to have the opportunity to directly bring the dye from the dark state, because it gives us the ideal control to see what is dependent on the proximity of the other molecule and what is independent of the proximity of the other molecule. This is what we get in between the UV and the green light. Okay. And so I think we would lose that if we go to the blue for the donor.
Thanks for the great talk. I was really fascinated about what you showed with the IDRs changing the on rate of the transcription factors or clear factors. And I was curious, what happens if you do the opposite? You add extra IDRs. Do you sequester away all these nuclear factors from the DNA, or can you increase the association?
Yeah, this is really the experiment we did with hypoxia-inducing factors, right? Just changing the IDR of one by the IDR of the other one, and the IDRs are different. We're able to bring the properties of binding of one to the other. Here we had a twofold change, in binding occupancy that is clearly not a change of the K off, that is a change of the K on because of the IDR. And so we can clearly change the dynamic of a factor. SP1, I didn't show this experiment. We can recreate a functional SP1 with a weak DNA binding domain that has no specificity for the cis-acting elements of SP1 and just putting the IDR of SP1. This is going to be sufficient for rescuing. And
if you go again, like you double again, does it get a point where they stop being able to bind DNA? Because in cancer, for instance, many of these nuclear factors have truncations and things, so they get extra IDRs attached to them.
So what we have seen in different mutants, whether you increase the DNA binding affinity of the folded DNA binding domain, which very often are a few mutations away from binding far more efficiently, or changing the IDR, is you get into a system where they bind very effectively to DNA or to chromatin, but to the point where they don't make it to the target anymore because they bind basically to the first sequence they see. And so the balance we are playing in cells all the time is, I want to get to my target. So I want to bind specifically to my target, but if I bind too strong for DNA or chromatin in general, I'm going to be lost in decoy sites, right? And so the keeping weak affinities for multiple things is really making sure that you only bind to the place where all the stars are aligned and where you have your multitude of factors that are going to be present together. So it's really a village, but the village is very specific, right? And so you create the specificity by this multitude of interactions, but weak interactions.
Okay, one last question.
Thanks for the talk. I have a pretty naive question, or maybe I missed it, regarding the PAPA.
Yeah.
Is it a FRET thing between the receiver and the sender?
No.
It has nothing to do with it, because in FRET, when you have the spectra emission of the donor overlapping the spectra excitation of the acceptor, it means that there is a FRET. So
it is not a transfer of energy, and the beauty of this system, why is it so helpful for us? It's because we are measuring the proximity of two dyes at the moment of photoconversion, right? So it's photoactivation as a function of the proximity of another dye. In FRET, you are emitting photons as a photon of a transfer in between the two dyes, and you lose a lot of energy in the process, right? You lose so much energy that we cannot make it work in our live cell systems. FRET doesn't work. We lose so many photons that I don't see single molecules at the end in cells. My budget of photons is typically 50 to 100 photons, per image to detect a molecule. So if you FRET, you lose so many that you don't detect anything anymore. Here, you photoconvert a dye with a green light, and it's only going to become fluorescent in red as a function of the proximity of another protein at the time of photoconversion. Once it's photoconverted, it's a full dye. It's going to live its life whether your molecule stays or not, actually. But it's going to stay fluorescent, and now I can image it as a single molecule and follow its life. So I am not measuring an interaction every time I see the molecule. I don't know if this molecule is still interacting. What I'm telling you is at the time I send the laser pulse to photoconvert, this molecule was in proximity to a green dye. And now I am following the life of this molecule, which is great because I can measure decays, right? I know they were interacting at time 0, and now I can see how they change their dynamic And now I know, because I have identified my peaks of diffusion, I know which one is who, and I see how this curve, of diffusion coefficients is changing over time as a function of dissociation, for example, of this complex.